Welcome everyone to my channel. This video is talking about the syllabus briefing of Stpm Biology Semester 3. So for this video, those who just finished your Sem 2 examination, you can watch this video and make your preparation yourself to enter your Semester 3 lives. And also you will know what you have to learn in Stpm Biology Semester 3. For this semester, you total have 6 chapters you have to learn like semester 1. But in this semester, you have 2 different themes you have to learn that are ecology and genetic. So these 2 are totally different themes, but it is actually the MPM put in this semester. So in this semester, for ecology part, you have to learn 3 chapters, and for genetic part, you have to learn also 3 chapters. So you can see that the chapters seem same as semester 1, but actually the quantity of content for this semester you have to learn is actually more than in semester 1 and semester 2. Because in each chapter you have to learn many things. Later I will explain the syllabus by chapters. So in this semester, ecology part is your first theme and genetic part is your second theme. So you have to learn ecology first. The first chapter is taxonomy and biodiversity. It seems that you had learned already in SPM, but also you have to learn in SPM, but in C15 and C16 maybe is new to you, ecology and selection and speciation. So these are the new things to you lah. I can say. For genetic, there are also 3 chapters. So C17, inheritance and genetic control, and this is the longest chapter. It is the longest chapter in this semester. And later you can see how long is it. You have to learn many things because actually it's 2 different topics. Inheritance and genetic control actually is two different topics, but MPM put in the 1 chapter. Inheritance and genetic control. And inheritance, I think in SPM you already learned about the Mendel. But in STPM, you not only learn Mendel, later you will see it. And for C18, gene technology, and C19, biotechnology, actually in SPM you also have learned about the genetic engineering. C18 is about genetic engineering. But in SPA you just learn about vocabulary briefing only, but in SPA you learn very detailed, and there are 1 chapter for it. And for C19 biotechnology is the application of biology lah. So for those who maybe you are interested to further studying biotechnology courses in degree, you can see this chapter. So in this chapter you will learn all the application using technology in biology field. So there are total 6 chapters in this semester you have to learn. Okay, for first chapter, chapter 14 is about taxonomy and biodiversity. So the first subtopic is about the taxonomy. So you have to learn what is meant by taxonomy first. So importance of taxonomy, and then the second topic is concept of species. Okay, you have to know what is species. Okay, the third topic is taxonomy hierarchy. So for briefing in this first subtopic, you have to know the concept first, and you have to know that in the world there are many organisms, right? So taxonomy is one of the classification. It helps the scientists to easier to classify all of the organism in the world. So you have to know one of the importance and concept of species. How do we determine species? Because we have to determine species, not determine simple only, or like a human, or human is 1 species. No, human actually has many different species, like have different many species. Also So for taxonomy hierarchy, there are classification of organism. How do we classify the organism in the world? How scientists classify? Because we do not classify by simple, no, chin chai lah. There are also the true way, the ways to classify the organism in the world. Okay, after you already learn the taxonomy, the concept, theory only, then you will learn about the diversity of organism. That is actually is 5 kingdom system. But why I just say 4 kingdom? Because another kingdom is called bacteria, yang bacteria punya. Yang bacteria punya. Bacteria actually you already learn in semester 1. So for this semester you don't need to learn lah, because you already learn in semester 1. So in this your syllabus you only learn 4 kingdom system. What of these 4 are there? So Protista, fungi, plantae, and animalia. So you have to know all of these, and no way you have to memorize it. Because actually, if you already learn for taxonomy hierarchy, you will know that there are like a pyramid one. The largest one is kingdom. Sorry. The largest one is kingdom. So there are 5 kingdom system. The Protista, fungi, plantae, and animalia, and another one is for bacteria. And there are many classification. The second one is phylum. But in your syllabus, you just only learn about phylum until phylum. But for some kingdom, you will need learn until class. Later you will learn. So phylum is the must. You will learn until phylum. One kingdom system for Protista, there are 3 phylum you have to learn. For fungi, fungi is the easiest one. You have only 1 phylum only. And plantae and animalia, plantae you already know, right? Plantae. Just this why not call plant? Plantae actually is plant, right? But in the taxonomy kingdom system, we not call plant, we call plantae. Same as animal. Animal we not call animal, we call animalia in the taxonomy system. So plantae actually has many. Already for plantae actually has about 5 phylum you have to learn. You have 5 phylum, and then animalia is the most one. You have to learn about 7 phylum. You have to learn, and each phylum what you have to learn about the characteristic. What are the characteristics of it? How do we separate? Why there are 5 phylums? Each phylum has their unique characteristic. What characteristic? And then this first one, and the second one is example species. What are the example of species under this phylum? You have to know. Only these 2. You seem these 2 are you 2 only, but the characteristic actually is very detailed, not just simple like plantae have chloroplast. No, not like that. Or animalia have 2 legs, 3 legs, 4 legs, no legs. No, not like that. You have to learn in detail one, and you have to give in scientific terms. Okay, so this is the second topic, and this is the longest part for this chapter. Longest part because there are many phylum you have to memorize it. Okay, and then the third one is biodiversity, but you have to learn in Malaysia, not in the world. The syllabus in Malaysia, you have to learn Malaysia. So you have to learn the 3 class level of biodiversity, and then the importance of biodiversity. And then the 4th topic is about threat to biodiversity. Apa ancaman? Bahasa Melayu kita cakap ancaman untuk biodiversity. What is the threat? The factor or threat. Macam ancaman untuk alam sekitar lah. Dia macam ni lah, tapi bukan yang kamu buat karangan SPM. SPM list out the factor threat to biodiversity. You just list or throw the rubbish into the sea, overfishing like that lah. No, you have to give in scientific terms. It seems easy. This topic is very easy. Like SPM, even 83% also can answer, but in STPM you have to answer in scientific way. You cannot answer like macam SPP3 punya student like that. And then also you not only learn factor, you have to learn the langkah untuk mengatasi ini. Okay, then the last topic is conservation, pemeliharaan of biodiversity. So there are two types, in situ and ex situ. You have to know what is meant by in situ and ex situ and the importance, their importance. Okay, so that's it overall for this chapter. So in this chapter, actually is easy topic lah. I can say it is easier chapter because you just memorize it. Memorize it. You have to all are memorized. It is very easier to understanding. This chapter is very easy to understand, actually. So this chapter has not too difficult at all. You have to like that lah. Okay. So that's end for this chapter. Okay. Why this like that? I think this is bug. I don't know. Okay. Sorry. Okay. Then the second chapter is about ecology. Ecology, I think maybe it's new to you, but never mind. You will learn at this STPM. Okay. So the first topic is about the level of ecological organization. So you have to know the level of ecology in the world. Actually, ecology, if you know the definition of ecology is the world environmental, right? Wherever environmental that have lived in organism, like the earth. One earth is also known as ecology. So there are many levels of ecology you have to learn. So hereditary in ecology, and then you have to learn interaction, biotic and abiotic factors. So you have to know what are biotic and abiotic factors, and you have to learn their interaction. Okay. Then the third one is type of biome, and then the fourth one is nature. You have to also learn the type of nature and their importance. Okay. Next is about the biogeochemical cycle. Okay, this one actually one of the example that you have to learn lah. The nitrogen cycle. Nitrogen cycle actually is SPM level. I don't know KSSM have learned or not, but KBSM learned. But in STPM, no. It does not indeed the syllabus. Maybe MPM anggap sudah belajar di SPM lah, so tak perlu belajar di STPM, I don't know. But no in the syllabus. But it is one of the example of biogeochemical cycle. So in SPM, you not learn nitrogen cycle, but you learn another 3 types of cycle that are carbon cycle, phosphorus cycle, and sulfur cycle. Some same macam nitrogen cycle saja. Dia macam nitrogen cycle, just the component are different. Nitrogen tukar kepada carbon, phosphorus, and sulfur like that. Okay, the third one is about the energy flow. Energy flow, this one I think your energy flow, I think this one macam SPM lah. If you have learned about the food chain and food web, I think not SPM lah. In primary school, I think actually already learned already food chain and food web. How the energy flow? As we know that there are producer, and then you have primary, and then you have a consumer, and primary consumer, secondary consumer, tertiary consumer, like that, right? And then the energy flow, how the energy flow from producer to consumer, and primary consumer, and then from primary consumer to secondary consumer, like that. So this is the intro of energy flow. So this is just intro only, not too detailed, but intro. So you not only learn food chain and food web, you also have to learn law of thermodynamics. Okay, the second topic is about the energy flow in tropical forests and lake. In SVA, you just learn a food chain and food web, right? Food chain and food web just a small part, small field only. But in tropical forests, it is very big area. There are many different types of animals and plants within that area. So you have to know how the energy flow happen in tropical forests and lake. The fourth one is about the population ecology. This is totally new things. You have to know the population ecology. So the first and second one is actually one topic is the exponential curve and logistic curve. These two type of curve, you have to know the curve shape. You have to know one first thing, you have to know the curve shape and the meaning, the description. What are these? The curve means, and also the labeling. You have to know all the curve, and from the curve you have to know the description. You have to know what does this curve tell us. Okay, the third topic is survivorship. There are also one type curve, one type of graph. Also, you have to know the meaning of graph. What does the graph tell us? Okay, then the last one is K and R strategies. What are the meaning? And then what are the example of species? And which of the example species belong to K strategies, or which of the species belong to R strategies? So this chapter, this topic actually is considered as hot lah, because many times that come out in passive. If you have seen passive later you learn this chapter, you see passive many years. I can say many years come out. So this is considered hot topic. Okay, the fifth one is carrying capacity. What is mean by carrying capacity? And then land, you have to learn sustainable development. Okay, and then the second one is factor limit population size. Okay, apa faktor menguangkan population size? As you know that why the population size cannot increase like that until infinity. Why cannot? Actually can, right? Why cannot? Because there are some factor. But if you see that actually for any population, even in human population, you see that it will like that one. Not like the straight line, straightforward until infinity. Cannot one. Actually cannot one. Even human also cannot. There are some factors limit the population size. What factor you have to know. Okay. Then this one is also the hot topic. This is the calculation question. Actually, I think you have also learned in SPM for like the density of organism, and then the percentage coverage. These are sampling method. All the formula you have to memorize it. You have to memorize it, because in examination no formula given at all. You have to memorize all, and this is the bonus that I can say is bonus for you lah. Even normally will come out in OBJ or structure. If come out in structure question, wah, awak untung lah. Structure question 7 atau 8 markah kan. Wah, 7 8 markah sudah dalam tangan lah. Ya, sekiranya awak ada hafal formula lah. Kalau awak tak ada hafal formula, tak pakai formula, memang rugi lah untuk awak. Ini untuk bonus kepada awak lah. Senang saja, very senang saja. Dia simple maths only. Tak ada maths macam math T, ada integration, differentiation, tak ada. Dia hanya simple maths saja. Awak just use the formula, sub value into it, you will get the answer. Okay, the second one is pattern of distribution. Okay. So overall for this chapter, not easy, not so easy, but can consider as easy also. Just you have to take more effort lah. Okay. So for this chapter, like that. Okay. The next chapter is the ecology part, last chapter, but for this semester is third chapter. There are selection and speciation. Okay. This is totally new thing, but the first subtopic is you are familiar with it. That is variation. So I don't talk about that lah. Because variation in SPM you already learn. So the same thing only. You SPM what you learn, you will learn in this topic only. Yeah, but yang penting ialah ini natural selection. So you have to know what is mean by selection first. Selection first. Yang saya cakap dalam kasar lah, untuk yang briefly, just in environment you will see that the organism need to survive one environment. The species will select the suitable characteristic or suitable phenotype. If you learn phenotype, genotype, you have to learn the terms lah. As if you already forget what is mean by genotype, phenotype, better you baca balik SPM Thus you have to, the species was choose the best phenotype that can make them to suitable for the environment. Like for example, like the rabbit color, fur color. Okay, there are white color and black color, and they have to survive at the hole. Lubang, mereka akan survive dalam lubang punya batu lubang punya. Okay, batu lubang yang awak tahu cahaya dia kan, the lightness of the hole is bright or dark. Dark light is dark light. So if the white rabbit live at the hole, is it safe or not? Kalau dia tinggal dalam batu lubang macam ni, untuk rabbit putih, dia akan selamatkan. Tak selamatkan. Yang predator yang predator akan tengok, wah sedapnya ada rabbit yang macam ni, kerana sangat terlalu macam mana cakap, terlalu easier to see it at that environment. So if the rabbit with this color, only with this color at that environment, in the future, the environment will select the rabbit fur color into the black. Only the black fur color rabbit can easier survive in the hole. White rabbit cannot, right? White rabbit, if you put the white rabbit in the hole, predator easier to see it and he will makan oleh predator. But the black is not easier to see. So in the past decade, very more need a long time. Very need to the long time baru dia boleh become the black fur rabbit color. Like that lah, dia concept macam. Tapi concept macam. So, but this one actually require very long times. Not 1 tahun 2 tahun boleh punya. Dia macam nak guna beberapa juta tahun punya. Need the billion or million of years to change the phenotype of the organism that can suitable survive in that environment. So the concept like this So natural selection. So there are 5 types of natural selection you have to learn So you have to learn. But for this three type is the most important one because they are graph also. Ada graph yang berbeza, and examination normally will keluar graph, then you have to know this which graph is responsible to that the selection. And these two one no graph, but just the explanation only. You have to know the explanation. Okay. Then the second one is artificial selection. This one is natural. Natural is yang semula jadi punya, original punya, yang environmental nak punya. So natural selection require very long time because need to change the phenotype is not very easy one. It need the very long time. But artificial selection is yang human buat punya. Human nak apa, dia akan jadi apa. So it is very short time. Require maybe 10 years, 2 years, 10 years, 20 years, it will already change. Not like the natural selection require million of years. So artificial selection it require only short time. So what are the ways that human can do it? Human have create about the human sperm bank, animal sperm bank, and gene bank. Use the genetic information of the organism, then the human can choose what they want. Like the vegetables, genetic change of the vegetable. Tomato want to become more juicy. What gene you have to add, or what gene you have to change. So according to the gene bank, like macam human also. If your wife and you want to get a child like Einstein, or atau you have to get a child like very handsome punya child, you can go to human sperm bank and get the genetic, get the sperm who are very handsome. Like your idol, K-pop idol star, maybe. So you will get the human sperm from your idol, then do the IVF, then you will get the child. You will get the baby, your child, like the handsome boy. Like that lah. This is just artificial selection. Okay. Next one is speciation. So selection topic is finished. Now is speciation. Okay, speciation is what? What is mean by speciation? You have to know and type of speciation. Normally, speciation is formation of species. Macam mana satu spesies boleh dijadikan? Macam mana nak jadi spesies? Actually, we want make one species or totally new species is very difficult. It is not easier one, but actually can. But there are the ways to have to know. So type of speciation, and then the second one is isolation. Isolate this and type of speciation, and then these four topics are the classification of speciation. Macam mana species boleh jadi? There are isolation, genetic drift, hybridization, and adaptive variation. You have to know all. Actually, I have to know all. What are they? And then there are also many classification about isolation. You have low pre-zygotic and post-zygotic isolation. Genetic drift, you have to know bottleneck effect. And then hybridization, you also have to know all lah. Semua yang ni hanya hafal lah. Boleh kata hafal lah, but you have to understand first lah what are the concept first. Okay, so this is all for this chapter. So this chapter actually moderate lah. I can say this chapter hardness are moderate because it is totally new except the first chapter. Except the first chapter only you have already learned in SPM. Only this chapter you learn in SPM, and others are totally new to you. So it is considered as moderate topic, moderate hardness lah. But also can, if you read carefully, understand carefully, and actually you can understand well. Okay, so this chapter is finished, and I will leave talk all the ecology chapters already. Next, I will proceed to genetic themes chapters. Okay. So chapter 17 is the first topic of genetic themes. So it is inheritance and genetic control. And you have to know this is the longest chapter. Paling panjang tu. Kerana it has 8 topics. Wow. You have to know 8 topics. And then each topic you have to learn many things. Actually, you have to learn many things. 8 topics. If you see the chapter 16, 14, 15, 16, only few topic only 3 until 4 only. Yang paling banyak I think 5. But this chapter you have to learn 8 topics. Very long. And normally your school teacher will require 1 month. Yang paling very fast. You have to require 1 month to learn only this chapter. Okay, so we are wasting time, so you have to know you have to get focused because this chapter very long and many things are new. Okay, yang pertama type of genetic codes within system Mendel. Ini kawan awak lah, SPM kawan awak sini saya tak mau cakap banyak-banyak lah kerana ini totally SPM punya. Totally SPM you have really learn yang Mendel punya. Mendel experiment, monohybrid inheritance, and dihybrid inheritance. You already know. So normally, even your school teacher will not take more time, waste your time at this topic because you already learn in SPM. Okay, then the second topic, yang ni topik yang kamu tak pernah belajar. Yang ni topik semua kebanyakan orang akan confuse dengan Mendel topik. Because in Mendel, if you know Mendel yang saya cakap bijak Mendel, if you know their genotype, if like the T, I consider T is gene T is tallest, the tallest, and T yang bigger, capital T is tall and small T is short. So if the organism for Mendel say that if the organism that has the hybridization genotype, like this hybrid genotype, it will present the characteristic will present as will present dominant one. Means that this organism is tall, not short. For Mendel dah cakap macam ni. Kalau ada gene like that, hybrid gene like that, it will present the dominant phenotype only. It will present dominant phenotype. It will not present the recessive phenotype, and it also will not present like medium punya. Yang Mendel dia cakap, nanti dia akan tall. Phenotype dia tall. So even though it has recessive genotype in it, but actually in the world, no, not all organisms follow this. Actually, there are many exceptions also. And then Mendel only say that one phenotype, one characteristic is controlled by one gene. Like this is one gene, right? No, who say? Actually, in the world, there are many organisms, one characteristic can control by many genes. And also, Mendel say that only one characteristic only has one dominant and one recessive. Dia cakap ada one dominant, only one recessive. And siapa cakap? Mendel dia cakap. Tapi in the world, no. There are many exception from the Mendel. So Mendel actually you know that Mendel is the father of genetics. He is the first people who founding the inheritance concept. But in the after many years, scientists make more research. Many scientists found that actually not all organisms follow Mendel. Not all organisms follow Mendel. So for the second topic is non-Mendelian inheritance. Yang inheritance yang tidak ikut Mendel law, totally tak ikut. So there are, you can see banyak kan? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 topics. 9 topics you have to know. And you have to know what are the difference between this man-made and Mendel. You have to know. You have totally to know. Because if you don't know, you just memorize in examination, exam come out. Ini Mendel atau tak Mendel. Awak pun tak tahu. Itulah. So you have to know the differences. So the first one is incomplete dominance, co-dominance. Incomplete dominance is the for saya cakap kasar briefly. I explain briefly only, like the TT in incomplete dominance, it will have another phenotype characteristic. For Mendel, we only have like TT, we will only have tall, like the tallest of plant, you only have tall and short. But in complete dominance, it will have another characteristic. So it is incomplete dominance. For co-dominance, there are the characteristic, they have more than one dominant allele. Like ABO system. ABO group. If you know that the A and B group, B, E are dominant, totally are dominant. That is why we have AB group, AB blood group. Co-dominance, and also multiple alleles. For one of the example, also the ABO blood group. Why? Because there are 3 alleles, right? Because in Mendel only 2 alleles. Either T, either capital T or short T. But in multiple alleles, there are more than 2 alleles. Maybe like ABO blood group, A allele, B allele, and O allele. And then a letter gene is dia macam Mendel, tapi kalau ada satu gene keluar, dia akan mati. Mendel tak ada mati, tapi dia akan mati. Yang ni letter gene. Then polygene other gene, one characteristic is controlled by many genes. Not only one gene, not only this TT. Maybe ada AAO, ada BB, ada CC. There are many genes only control the tallest. Hanya control ketinggian sahaja. Control ketinggian hanya ada 4 gene ni control the ketinggian satu organism. Ini polygene lah. Yang lain link gene and sex link gene ini sama sahaja. Actually sex link gene is involved in sex chromosome. Link gene yang lain autosome chromosome. Epitasis and pedigree analysis dia macam polygene tapi ada sedikit berbeza. Epitasis is when there are the gene occur at the genotype, it will totally change the characteristic. Like something like that. Dia macam polygene. Polygene adalah satu gene, banyak gene control satu characteristic. Ini pun sama. Tapi kalau jika ada satu gene keluar, dia akan totally change the characteristic. Even though yang lain gene semua dominant. Saya cakap example lah. AA, BB, CC, and DD. Dan macam ini, kalau orang yang ada gene ini, dia akan keluar skin color, dia akan white. Dia akan ada white skin color. But if let's say ini yang dominant lah. Let's say ini just an example and then the recessive one is the recessive of skin color maybe is black. Black yang macam mana, maybe yang small one, all small. But if the gene content, if the DD gene become DD, yang become hybrid one, even though all of the yang ini AA, BB, CC, all are the capital, but it will totally change the another phenotype, maybe girl. it will totally change to brown color, brown skin color. That people will get brown skin color, even though even though it's dominant or recessive, even though like that, like that also. But got one this gene, it will become brown color. This is called as epitasis. And then the last one is we degree analysis. Ini macam keluarga punya, macam inheritance but you have to know the disease from the family. If you already know that this disease is inheritance disease, you can know that which your father, mother, grandmother, grandmother who get this disease until you get. So you can make a prevention for this. So these are the 9 topics for non-Mendelian inheritance. Actually, is complex, hard. I can say hard topic because normally many people confuse. Not only between this topic and also between the Mendel one. Actually, many people will confuse like that. So you have to take more focus, effort for this, and better you take a notes. Okay, so this is just the second topic. This is only the second topic. I already say this chapter has 8 topics. Only is the second one. Okay, next, third topic is genetic mapping. Okay, so genetic mapping actually is easier topic. No, not say not to focus, but it's easier. It is also just learn the your gene only. Then the fourth topic is population genetic and Hardy-Weinberg equation. This one is calculation. Sem 3 banyak calculation. Ecology ada calculation. Ini chapter ada calculation. You have to know the equation. Equation only easy only hanya 2 equations saja. A tambah B equal to 1. A square plus 2AB plus B square equal 1. Senang je. Takkan tak boleh hafal. A plus B equal to 1. A square plus 2AB plus 2B square equal to 1. Just you have to know what is A, what is B. And then also what is A square, what is 2AB, and what is B square. You have to know that. If you already know, then you can do this calculation. Senang je. Also, I can say that bonus to you. It's bonus to you if come out. Awak untung lah. Okay. Next is the genetic control topic. Yang 7.1 until 7.4 are the inheritance. Next, begin 17.5 is DNA replication, is about the genetic control. Okay, DNA replication. Okay, as if you have learned in SPM, I don't know you got know or not, because in SPM you have no learn about mitosis, right? But before mitosis you have S stage, because in the cell division stage, mitosis is just one of the stage. Actually you have another stage, right? S1, then beginner stage, and then S2, like that one. And one of the stage you have DNA replication terms, right, in SPM. I don't know you until now. I don't know you just memorize or not, but maybe you already forget. Never mind. But I think you have to know that DNA replication terms. So maybe in SPM you blah what is DNA replication? Never mind. And then in SPM you will know. So, but before you go to the mechanism of the DNA replication, you have to know the history of DNA replication, sejarah DNA replication. So, you have to know experiment proof DNA as genetic materials. Okay, so yang sejarah dia kalau example mula-mula saintis mereka langsung tak tahu DNA as genetic materials. Mereka hanya tahu nucleus carry genetic. information, genetic materials lah. Mereka hanya tahu ini saja. Kan awak sel awak kan ada satu yang paling besar punya nukleus kan. Tapi mereka tak tahu yang DNA carry genetic materials. Mereka tahu nukleus carry saja. Karena nukleus bukan hanya ada DNA. Nukleus ada DNA, dia ada protein juga. Dia ada karbohidrat juga. Dia ada RNA juga. Tak. Tapi yang mana satu yang carry genetic information, saintis tak tahu. Yang tu mula-mula punya saintis mereka langsung tak tahu. So the scientists want to know which component carry genetic information, genetic material. They do experiment. So there are 3 experiments you have to know. And also 3 experiments. Also you have to know 3 scientists name. But no, 5 scientists name. The first experiment only 1 scientist. Second and third has 2 scientists. You have to know the names and then the procedure of experiment and then the result of the experiment and also the theory of the experiment, conclusion of the experiment. So yang paling penting, yang name, nama punya scientist you have to know, and then the procedure of the experiment. Macam mana mereka buat eksperimen ini. Dan next is the result of the experiment. Result of the experiment you have to know. Okay, then you have to know the conclusion. Apa conclusion mereka buat? Kenapa nak sampai ada 3 eksperimen? Actually one experiment already enough if you want to prove as DNA as activity. But the history, yang previous scientist mereka buat sampai 3 eksperimen. So you have to know these 4: name, procedure, result, and conclusion. Okay. The second one is model of DNA replication. Okay. After scientists, yang mula-mula scientists, okay, mereka sudah dapat DNA actually is the genetic material. But macam mana DNA replicate lah? Replicate awak tahu kan? Copy. Kalau kami cakap normal term is copy. Copy and paste. Tapi saintis ada problem lah. Saya sebagai saintis macam mana DNA replicate lah? Siapa boleh bagi tahu? Tak tahu. Saintis pun tak tahu. So there are one scientist, I think if I not mistaken, for someone actually have learned core bricks and block. Not bricks and block. Siapa? DNA model punya scientist. Let me think first. DNA scientist. Watson. Ini Watson and Crick. Ni scientist. Yang ni scientist awal sempat ada jumpa kan? Dia yang propose DNA model kan? But Watson and Crick not only DNA model, it also has make a model, propose a model of DNA replication. Dia ada bagi 3 model. Yang maybe macam 3 possibility lah. 3 possibility that DNA can replicate in the cells. Yang dia yang bagi. Watson and Crick bagi. So you have to know all the 3 model you have to know. Macam mana dia replicate. Yang bukan macam mana, yang overall saja. Yang 3 different model of DNA replication. Okay. After yang sampai situ saja. Watson and Crick dia buat sampai saja. Dia tak ada buat proof. Dia mungkin Watson and Crick anggap ketiga ini pun akan berlaku dalam sel kita. The scientist thinks that these 3 models will occur in the DNA replication in the cells. But in the future, after that, ada scientists dia cakap tak apa, bukan. Mesti ada hanya satu. Only has 1 replication model only in this DNA replication. So there are another experiment again. The 4th experiment. There are another experiment. This is 3 experiment. Then you have to another 1 experiment to prove the DNA replication. Which model? 1, 2, 3. 1, 2, 3. First, second, third model. Which model say model actually is the DNA replication model. You also same as these 3 experiment, you have to learn the name of scientists, siapa yang buat eksperimen ni, also the procedure, macam mana dia buat, result, the keputusannya, and then the conclusion, what are the conclusion he makes the result from the result, and then you have to know the conclusion, and then the conclusion are which model are the conclusion that actually only the ones for DNA replication. Okay, after buat experiment, okay, then another scientist only, wah banyak scientist wah tak, saya pun tak mau cakap apa lah, maybe you hate him lah, wah ini orang ni cakap saya susah loh, pas saya nak belajar semua ni ini scientist lah, mereka scientist yang buat punya, mereka yang founder punya, dan then the finally is DNA replication mechanism. So you already know the model, then you have to know how the DNA replicate. You have to know lah. Okay, so this mechanism also very important, and then there are many enzymes named. There are many enzymes play a role in this mechanism. Have many enzymes. Like I give the example that DNA polymerase. Okay, DNA polymerase 3. There ada banyak DNA polymerase 1, DNA polymerase 3. You have to know all, and you have to differentiate it. And in the examination, you have to write carefully. If you just only write DNA polymerase, salah, totally salah. If you add that, or if you write salah, also totally salah. You have to mention 1 or 3. So you have to know the role of all the enzyme in DNA replication mechanism. So this one topic I think is hard topic. It's hard topic because a bit too detailed for you. Maybe it's too detailed for you. So good luck. Take more effort for this chapter. So I encourage you to read the Campbell book. Is more detailed for you, because if you read Oxford, I think blur lah. Confirm blur. Our confirm blur. If you just read Oxford, our confirm blur. Okay. Next is gene expression. Okay. So the first copy is you have to know gene concept first. Apa itu gene? Apa itu gene? In the DNA replication, you know that DNA is genetic material. Then now you have to know gene concept. Apa itu gene? How do we define gene first? So there are another experiment. Ini yang eksperimen kelima, Bieder and Tetten experiment. Ini scientist dan punya. Bieder and Tetten experiment, dia prove gene concept. Mereka buat eksperimen ni, then found the gene concept for us until now. Okay. And then the second one is genetic code. Okay, what are the mean of genetic code? Okay. Then the third and fourth one is very important one, is mechanism process, transcription and translation process. So actually, if you in semester 1, if you learn the DNA topic, nucleic acid, RNA function, actually, if you know that mRNA and tRNA actually function, you have know that mRNA function is play the role in transcription, tRNA function is play the role in translation. But many people ask, apa itu transcription, apa itu translation? Many people ask me. Tak apa, di semester 3 awak akan tahu apa itu transcription, apa itu translation. Yang paling penting, yang general punya yang mereka ialah proses untuk sintesis protein synthesis to produce protein, because our bodies can produce protein, right? And our body can produce non-essential protein. If macam mana protein, betul ke? Maybe salah lah. But if you know that our protein can produce protein, but we have essential protein and non-essential protein, but I already forget which one produced by body, which one are not produced by body, but I already forget. But actually, you have to know. You don't need to know. I just want to mention you. So this is just protein synthesis process, because we need these two process to synthesis the protein for our body. Okay, so this is fifth and sixth topic. Okay, next one is about the regulation of gene expression. If you know, the sixth topic is gene expression. Gene expression. Now you have to learn regulation, but for your syllabus, you just learn prokaryote only. You not learn eukaryote. Prokaryote lah bacteria. Bacteria is one type of prokaryote, but you not learn eukaryote. Like human and plant, you not learn. Okay, so you have to learn the operant system, and then you have to must learn the operant system, and then the second and third is the mechanism of lead operant system and mechanism of tryptophan operant. These two are operant, but there are differences. You have to know that what are the mechanism of lead operant system and mechanism of tryptophan operant, and you have to know the structure of operant system, the main structure, and also the lead operant system and tryptophan system also have the different structure also. You have to learn. Okay, for last topic, finally last topic lah. This mutation actually in SPM you actually learn. So gene mutation actually SPM learn. I don't need to talk too much. Chromosomal mutation, actually chromosomal mutation have two right. That is chromosomal structure, 4 types SPM learn, and chromosomal mutation change in chromosome number also SPM learn. So this topic actually just a revision topic lah. But for topic for maybe have add some detail lah. Ada tambah satu detail, sikit detail, and you have to know the example of species, example of disease. Especially for this, like Down syndrome, Turner syndrome, Klinefelter syndrome, XO syndrome. Inilah semua lah macam ni. Macam ni chromosomal number punya. Okay, and then you have to know the symptom lah. What are the symptom of Turner syndrome, Down syndrome? All of the syndrome you have to know. So that's it for all together for these chapters. So this chapter actually, I already say, very long. Have 8 subtopics, 8 topics you have to learn, and then many topics are very new to you, and then you have easier to make a confused. So you have to take more effort lah. Ini lah, focus in this chapter. Memang kena fokus. Tak mau tak fokus, kerana awak pun tahu 8 topics. So exam pun akan keluar banyak question dalam chapter ini. You have to take alert lah. Okay, next is about chapter 5 to chapter 18, gene technology. Actually, it is genetic engineering, mechanism of genetic engineering yang paling penting. Mechanism of genetic engineering. I think mechanism of genetic engineering in SPM you already learn, but in SPM you just learn the yang muka-muka surface only. In STPM you will learn very detailed. So there are 5 stages. 5 stages you have to learn. If you learn SPM dia macam ni kan. SPM ada plasmid, then ada gene of interest. Dan ada enzim, ada satu enzim cut down, ada satu enzim cut down. Lepas tu dia gene of interest, mereka combine, lepas tu awak akan dapat ada kombinan punya lah plasmid. Lepas tu awak enter bacteria, masuk dalam balik bacteria, then undergo replication process, okay, finally finish. Yang ni SPM punya. Tapi in SPM awak tak akan learn macam ni lah. In SPM apa 5 stage dia? Yang 5 stage dia, the first stage is ni lah, removal of gene from donor cell DNA to plasmid. How how the gene are removed from the cell DNA and the plasmid. Okay, then the second one is placing the gene into a vector. Okay, you have to place the gene into a vector. Macam mana? Okay, then the third one is into the vector and then form this one recombinant plasmid. And then the third one is transformation process. Transformation process is actually put the plasmid into the bacteria back. Okay, then the fourth one is the process that you have not learned is screening and select the transformed bacteria. We have to confirm that all of the bacteria in the culture are recombinant bacteria. They already have the recombinant gene at that. You have to confirm that because we cannot. Because if you not do this process, you directly do the cloning process of amplification. Maybe they are the culture. Maybe got the bacteria that do not have the recombinant. You have to do the screening and select process first. Baru buat cloning process. So these are the 5 main stage. Macam explain, memang macam explain just in detail form. So this is the first, second, then third, and then third. The fourth is screening process. You have to screen and select, and then the 5 is cloning process. Cloning produce very large amount of bacteria. Okay, and then the second and third is genomic library and cDNA library. Actually, not saying not important, but you have to know also because exam also maybe will come up. Genomic library and cDNA library just a memorization that you have to know. Okay, then the last chapter, biotechnology. So this is the chapter that actually is interesting chapters for especially for those who are interested to further study the biotechnology degree courses in university. So there are 9 applications. Wow, memang 9 application you have to know. So the first, but yang saya cakap yang penting the most important is human growth hormone, insulin, gene therapy, DNA fingerprinting, and genetic screening. These 1, 2, 3, 4. These 5 are the important one. Memang important punya. Yang these 2 human growth hormone yang ni akan guna genetic engineering. Dia akan guna balik genetic engineering, dan gene therapy, DNA fingerprinting, genetic screening are totally new. Dia akan ada new application for you. You have to know. You have to know also. And for food and beverage production is cheese production, milk production, wine production, and beer production. But until now, tak pernah keluar lah. All section tak pernah keluar lah. Tapi saya I cannot promise that it will never come out in examination, but because in syllabus have. If you see the syllabus have, but tak pernah keluar lagi, MPM tak pernah keluar lagi. So I cannot say you cannot read. You also have to read also. And for this agriculture ada pernah keluar, tapi sikit saja, also have to read. And for this biosensor and oil decomposing bacteria keluar dalam OBJ lah. Biosensor actually is enzyme technology. If you learn enzyme technology yang Sem1 chapter 4, you should learn like that lah, biosensor. And then oil decomposing bacteria actually is just senang je, tak susah sangat. Yang ni senang je. Okay, so that's all for this semester syllabus briefing. So actually this chapter interesting chapter and not too hard, moderate hardness only. Yang paling hard punya adalah chapter 17. Yang itu paling hard, paling the hardest chapter, yang chapter 17. So that's all for me in this video presentation about the semester 3 syllabus briefing and I hope that everyone who watch until end, you can scan this QR code, or you can click the link below the comment, below the info that I will put. Just survey lah, simple survey only. I want to see that what are the feedback that you made after you see my video. So that's all for me. Okay, thank you. Bye.